Okay, hello everyone. Thank you for tuning in today to view my presentation, and I would like to thank Labroots for the opportunity to present my work, and I'm really excited to be a part of this online conference. It seemed quite exciting, and there are a lot of great presentations set up. So I'm going to take you through the research that I've been doing in the lab. So my name is Rachel Lackie, and I'm a PhD candidate at the University of Western Ontario, and I work in the laboratory of Dr. Marco Prado at Robarts Research Institute. So the project I'm going to be talking to you about today is looking at a particular protein and its sort of involvement in brain cell resilience or survival during aging. So the title of my talk today is Investigating the Roles of the Hsp90 co-chaperone STI1 in Neuronal Resilience During Aging. So first off, what is stress-inducible phosphoprotein 1? So since it's a bit of a mouthful, I'll keep it down to calling it STI1. So STI1 is a protein that is kind of referred to as sort of a moonlighting protein. So it has two dual functions respective to its location in regards to the cell. So inside the cell, it is known as a co-chaperone. So basically what this means is it helps out particular chaperone proteins that are important for protein quality control and making a immature peptide into a functional protein. So STI1 was originally identified for its function as a co-chaperone in yeast, and that it particularly is elevated in cells that are under different conditions of cellular stress. So there are different domains of STI1 that interact with these co-chaperones. And basically, it's a flexible protein that coordinates the functions of these proteins. Now, STI1 can also have very different activities outside of the cell. So here it interacts with the cellular prion protein. And this has a number of beneficial effects for the cell, which I'll get into the next slide. So basically, recently our lab developed an STI1 knockout line, as most of the literature up until this point regarding STI1 has been done in yeast or C. elegans worm models. So our lab deleted STI1 in mice, and it was found that this was embryonically lethal. And basically, it was alluding to the fact that STI1 is really important for development and survival. And interestingly, there is about a 98% sequence homology between mouse and human STI1. So then we looked at some human data sets, and it was also revealed that loss of function mutations in STI1 are not tolerated. So this is really highlighting the fact that STI1 is really important for mammalian function, or mammalian survival, and basically for life. So just to further elaborate upon STI1's functions, so like I mentioned, STI1 can be secreted by cells, different types of glial cells, or it's also expressed all throughout the body. So basically, STI1 interacts with cellular prion protein. Here it's denoted as being PRPC, and STI1 highlighted sort of in orange. And basically, this extracellular interaction leads to a number of neurotrophic signaling effects via calcium signaling. And basically, some examples of this are increased cell proliferation, neuronal differentiation, protein synthesis, all basically good things for cells. And then, like I mentioned, inside of the cell, STI1 was really critical for the coordination of Hsp70 and 90. So alone, these proteins cannot interact with each other, Hsp70 and 90. So you need STI1 to allow for these proteins to physically interact. So STI1 here being shown in pink, Hsp90 in green, and Hsp70 or HSC70 in blue. And basically STI1 is just acting as a client recruiter. So it allows the client that is currently being bound by Hsp70 to transfer to Hsp90. So this immature sort of client or protein, as I'm referring to it as, can then be folded through this machinery into a functional protein. So as I mentioned, deleting STI1 in mice was embryonically lethal. And essentially what our lab had shown with these mice is that basically not having STI1 really makes cells unhappy. You have higher amounts of cell death. This micro CT analysis here in the top right panel is showing that you have improper development of these embryos, which is essentially what's leading to death. And interestingly, when we took tissue from these embryos, was that we saw a decrease in the protein expression of a number of HSP90 client proteins. So all in all, not having STI1 really stresses out cells, and you need STI1 for viability in life. So me being a neuroscientist, sort of my interest was, what is really the functions for STI1 in brain, or how important is it? Because a lot of this work up until this point has been done either in vitro, looking in neuron cultures, or most of the work being done in yeast. So here, in this image here, was taken from the Allen Brain Atlas for an in situ hybridization for STI1. So basically you can see within the brain, it's really highly expressed, particularly in neurons, but you see it's really highly expressed in the hippocampus, the cortex, so and so. But as I mentioned, a lot of work about STI1 up until this point has been done in vitro. So as I mentioned, STI1 can be secreted, and glial cells within the brain are known to secrete STI1, such as astrocytes and microglia. And it's this secreted STI1 that is really important for keeping neurons viable and actually protecting them against a number of different cellular insults, such as different chemical stressors, environmental stressors, even protein aggregates. Having more STI1 or STI1 in the extracellular media really protects and reduces the amount of cell death. And then finally, more so looking at the clinical literature, it was shown that mothers of children with autism actually have higher levels of autoantibodies against STI1. So then this group here basically treated rodents with autoantibodies against STI1 and other markers that were predicted to be involved in autism. And really this led to a lot of changes in neurodevelopment of these rodents. So changes in brain volume and cell proliferation, which really contributed to the developmental delay that you see in autism. So as I mentioned several times, up until this point, a lot of work has just been done in vitro with regards to STI1. So we want to know what's going on in vivo in the brain, particularly during aging, when neurons are really vulnerable. So this is our main hypothesis for this work that I'll be presenting today. So basically, we hypothesize that normal expression of STI1 is necessary for neuronal resilience in vivo. So as I mentioned, the problem that we've had up until this point is that knockout for STI1 is embryonically lethal. So this is really, at this point, halted our ability to study STI1 in vivo, since getting rid of it is lethal. So what we did was we generated a hypomorphic allele mouse line, is what we call it. And basically, we're just using Cre-Lox-P recombination to delete a part of STI1. So here we're deleting exons 2 and 3. And just this schematic here, so this is just normal full-length STI1 gene, mRNA, and protein. And basically what we did was we deleted exons 2 and 3, which corresponded to a deletion of the N-terminal TPR1 domain. And this domain has been suggested to be redundant within the literature, as this domain, TPR1, interacts with Hsp70, but as does TPR2B. So basically this deletion we're truncating the protein, we're deleting this TPR1 domain, but because of this deletion, it led to the generation of a new initiation codon, which turned out to be quite inefficient. So taking brain tissue from these mice, we see that there's a significant reduction in STI1 protein levels. So just this Western blot here is showing wild type, so just normal controls, het, meaning heterozygous mutants, so meaning they're heterozygous for this allele, and then delta TPR is referring to our homozygous mutants, so both lacking this TPR1 domain. So essentially what we're seeing is in these homozygous mutants about an 80% reduction in protein levels, and then a corresponding decrease in protein molecular weight, since we are deleting a domain. So interestingly, despite reducing STI1 protein levels by about 80%, these mice do survive, which is great, so we could further characterize them and further study the roles of STI1 in the brain of living adult mice. So basically, things that we were noticing off the bat with this mouse model was that the survival earlier on in life, so just after being born, there's there was about a 15% amount of these homozygous mice being born. So it was particularly, or with normal Mendelian frequency, you would expect 25%, but we were getting about 15% of these homozygous mutants. And one thing that we particularly notice about these homozygous mutants is that they're a lot smaller and leaner than their heterozygous and wild type litter mates. So it was just that these mice were smaller and there's competition amongst pups, which is what was leading to death early on in life. As you see that their survival as they get older is pretty much just stays the same and flat as you see in wild type and heterozygous mutants. So other phenotypic characterization of these mice revealed that they're actually metabolically hyperactive. So they have increased oxygen consumption and CO2 production in both the light and dark cycles. And then this was also corresponding to increased locomotor activity. But other than these findings being leaner and being metabolically hyperactive, there were no gross phenotypes or health problems with these mice. So this allowed us to further study STI1 in vivo. So as I mentioned, STI1 is really critical for the coordination of an immature client between Hsp70 and 90. So we wanted to see if there were any consequences of reducing STI1 on this machinery. So this is just a schematic showing how STI1 is involved in the chaperone cycle in bringing a client, and then just a number of other co-chaperones and accessory proteins that are involved in this machinery that we also will look at. So first off, by reducing STI1 levels by 80%, we saw that this had no effect on the amount of mRNA or protein of Hsp70 or Hsp90. We also ended up finding, which I will show below, is that there was a reduced expression of a subset of Hsp90 accessory proteins or other immunophilins that were involved in protein folding. Classical Hsp90 co-chaperones, such as AHA1, CDC37, P23, and FKBP51 were not affected by reduced STI levels. But as I mentioned, we see a subset of Hsp90 accessory proteins that were reduced when STI1 levels were also significantly reduced. So again, just showing wild type heterozygous and homozygous mutants. And interestingly, we see CHIP, which is actually a protein that is in competition with STI1 for the interaction with Hsp70 and 90. STI1 promotes the folding of peptides or unfolded proteins into functional proteins, whereas CHIP is more so, it's an E3 ubiquitin lyase, so it promotes the degradation of target proteins. So interestingly, we see that this CHIP protein is reduced by about 50% in both our heterozygous and homozygous mutants. And then other peptidyl-prolyl isomerases, such as PIN1 and SEPA, were also reduced in our heterozygous and homozygous mutants. So next, we wanted to investigate whether client proteins of this Hsp90 machinery were affected by reduced levels of STI1. So a classical client of this machinery is steroid hormone receptors, or glucocorticoid receptor is sort of the best studied. And we see again in our heterozygous and homozygous STI1 mutants that there is a significant reduction in the protein levels at glucocorticoid receptor. We also looked at other clients of this machinery, such as total tau and GRK2, and again, in both our heterozygous and homozygous DUS2PR1 STI1 mutants, there's a significant reduction in levels of these proteins. So basically, this interesting finding of both heterozygous and homozygous mutants having reduced expression is we're thinking that STI1 could be acting in a dominant negative effect. So just having one mutant hypomorphic allele is affecting the wild type allele, and is really compromising STI1's activity. So we wanted to determine if the findings of disturbed Hsp90 accessory proteins or client proteins were due to deletion of the TPR1 domain, or if it was due to reduced levels of STI1. So using the CRISPR-Cas9 machinery, or the CRISPR-Cas9 technique, we developed an STI1 knockout neuronal cell line using SN56 cells, and we looked at the same HSP90 accessory proteins and client proteins, and essentially we see the same findings, that having, in this case, no STI1, you have reduced expression of clients such as glucocorticoid receptor, but that we also, again, are seeing reduced expression of PIN1, CHIP, and SEPA. And most importantly, in either the third bar of these graphs or the far side of the Western blot, that you see when we retransfect in STI1, that this rescues the reduction in these client or accessory proteins. So this is really showing that our mouse line is basically an STI1 loss of function. So taking all of this proteostasis work all together, we wanted to really know what were the consequences of reduced STI1 expression on the aging brain in mice. So after some preliminary work, we focused our analyses on the hippocampus, which is a brain region which is particularly vulnerable in as you age or different types of stressors. So one thing that we were noticing was some structural differences within the hippocampus of old mice. So first, we characterized younger mice. I should mention that most of these Western blots that I had just previously shown were done in aged mice, mice being around 15 to 18 months of age, but we saw similar findings when we took tissue from younger mice. But here, this data I'm going to show here, first we're showing younger mice. So we focused in on the CA3 region of the hippocampus, just for what I'm showing here today. Because this is a subregion of the hippocampus, which is particularly vulnerable as mice age and in disease, neurodegenerative diseases, or during stress. And in young mice, we see sort of a thinning going on in our homozygous delta TPR1 mutants, but nothing significant. But when we look at our 15 to 18-month-old STI1 mutant mice, we see a significant loss of neurons within this CA3 region of the hippocampus, which is showing a consequence of having reduced STI1 is really affecting the integrity or the viability of these neurons. So then we wanted to take things sort of to a larger scale, and we collaborated with a group at Sick Kids in Toronto, and we gave them fixed mouse brains from our STI1 mutant mice, and they performed volumetric analyses using 7 Tesla MRI. And essentially just what this data is showing here, so the left panel being wild type versus heterozygous STI1 mutants, and then on the right being wild type versus our delta TPR1 homozygous mutants. And the most striking findings, of course, being in the homozygous mutants, we're seeing an increase in brain volume, which was not necessarily expected because we saw that these mice were smaller. But if you think back, it was shown in the rodents that had autoantibodies against STI1 that it did increase brain volume in these mice. So just sort of match with the literature. But interestingly, we strikingly see really something special about the hippocampus, so that when you have less STI1, this is one of the only brain regions that had decreased volume across the whole brain in these STI1 mutants. And interestingly, we saw increased volume in the cerebellum and the brainstem, but for now, just for the purpose of this talk, just going to focus on the hippocampus. So because we did 7 Tesla MRI, and we were able to, with different segmentation analyses, actually analyze the volume of particular subfields of the hippocampus, including the dentate gyrus, CA1, CA3, all those different layers within the hippocampus. So strikingly, like I mentioned, what we find is that hippocampal volume is reduced in our homozygous STI1 mutants, but it's particularly we see significant loss of volume within the dentate gyrus of the hippocampus, and also a tendency for the CA3 to be reduced. So just to clarify, all of these MRI analyses were done in aged 14-month-old delta TPR1 control mice, them being control littermates, of course. So again, we wanted to know we're significantly reducing the volume of the hippocampus, and this brain region is really important for, of course, learning and memory. So are there any consequences for learning and memory when we have reduced hippocampal volume and reduced STI1? So within the literature, it has been constantly shown that damage or reduced neurons within the hippocampus in the CA3 region affects spatial memory. So just practically damaging this region, leads to spatial memory deficits. And then it's also been shown, even within Alzheimer's disease patients, that there is a particular loss of these neurons. So then what we did, just as a simple probe for learning and memory in these mice, was we trained them over 4 days in a whole, like a tank of water, to learn where a particular platform within, this pool was. This platform is semi-opaque, so they can't easily see it, but they learn to find it as there are different spatial cues within a room. So over 4 days, they're trained to find this target or this platform, and particular things that we look at is if they take how long it takes them over those 4 days to find the platform, the distance traveled to the platform, and their sort of mean speed within the pool as they're swimming. So this is referred to as the Morris water maze task. And basically, we do see, as there's a reduction in latency and distance, we do see that the mice learn over the 4 days where the platform is, so what is referred to as the target quadrant. So that's 4 days of training. And then on the 5th day, we put the mice in the pool of water, but there is no longer a platform. So basically, this is referred to as the probe trial. And essentially, we're just assessing whether or not the mice remember where the platform is. So this is referred to as the target quadrant, shown in my little cartoon here. And then we also analyze the % time spent in the other quadrants respective to the target quadrant. And despite seeing that these mice learn, they actually, within the probe trial, they have deficits in reference spatial memory. So this is really showing that there is a consequence of reduced hippocampal volume and STI1 on spatial memory. So just to summarize what I've shown you here today is that we generated a hypomorphic STI1 allele mouse, which allowed us to study the functions of this protein in vivo in mice, particularly as they age. And we saw that there were a number of consequences of reduced STI1, particularly that we see reduced neuronal resilience as mice age, particularly in the hippocampus, and this contributed to a spatial memory deficit. And our findings that they were reduced HSP90 accessory proteins in clients suggest that STI1 may be acting as a key regulatory node of the HSP90 machinery. So some of our future directions are trying to understand whether or not it's particular cell types that are contributing to neuronal resilience as mice age, as STI1 is expressed highly in most cell types. But the issue, of course, with this mouse line was that this was a global knockout. It was everywhere. So we're trying to determine if it's cell autonomous or non-cell autonomous mechanisms. So STI1 in neurons, or particularly maybe STI1 in astrocytes or other glial cells, that is important for neuronal resilience, as it was shown in vitro that it's the secreted STI1 by glial cells that keeps neurons healthy. So stay tuned for that. But for now, this ends my talk for today. So I would really like to thank my lab. There's a number of us, as you can see in this photo here. I'd really like to thank Dr. Marco Vanya, Dr. Marco and Dr. Vanya Prado, my supervisors, for all their help and support throughout this project, and all the different funding agencies. And of course, our collaborators, Dr. Jason Lurch and Lily from Sick Kids with their MRI expertise and analyses, and of course our funding agencies. And I'd really like to thank again Labroots for this opportunity to present my work. And I hope you learned something about STI1 in the brain. And thanks for tuning in.